you go. This meeting is being recorded. So, hi everyone. My name is Tracy Steinricken. I'm the president of the Australasian Mycorrhizal Society. Thank you for joining us for today's July virtual seminar. Before we begin, I'd like to acknowledge the traditional owners of the lands around Australia, New Zealand, and around the world where we are all meeting today. I'm speaking to you from just south of Maiwar, the Brisbane River, in Queensland. This is the traditional homelands of the Turrbal and Yagara people, the custodians of this land. I pay my respects to their elders, past, present, and emerging. Our presenter today is Professor Jeff Powell. I was fortunate enough to have Jeff as my PhD advisor, and he is also a past vice president of our society. Jeff is an ARC Future Fellow at Western Sydney University's Hawkesbury Institute for the Environment, where he has been studying interactions among plants, fungi, and soils since 2011. Before that, he did his PhD at the University of Guelph in Canada, where he studied the effects of genetically modified crops on soil ecosystems and turned an impressive number of non-significant results into a thesis. In between, he was a postdoc at the Freie Universität Berlin, not Brisbane, where he managed to stay productive despite the city's fantastic beer gardens. Today, Jeff will be presenting his talk titled Switching Partners, the Roles and Relationships of Dual Mycorrhizal Systems for Eucalypts. He will be sharing evidence that suggests the occurrence of arbuscular and ectomycorrhizal fungi in eucalypt woodlands is driven more by the health of the broader ecosystem than by the needs of their host. If you'd like to ask a question or post a comment for Jeff, you can do so by clicking on the Q&A function at the bottom of your screen, and Jeff will answer your questions at the end of his presentation. Then at the conclusion of the Q&A session and the end of the seminar, please just take a moment to answer the short survey to help us improve delivery of the series. Thank you so much, Jeff. Take it away.
Great, thanks, Tracy. Let me just switch slides there, yeah. Thanks very much for the invitation to give a talk, Tracy, and thanks very much for the nice introduction. I was gonna say, I was fortunate to have Tracy, to be an advisor of Tracy during her PhD. She made it very easy. Yeah, so today I'm going to be talking about some work that we've been doing over the past almost a decade, where we've been looking at arbuscular and ectomycorrhizal fungi in some eucalypt woodlands in the Sydney region. Just a brief outline of what I'm going to talk about. I'm going to focus on, I'm going to give a very, very brief overview of ectomycorrhizal and arbuscular mycorrhizal associations, and get into it a bit on the context dependency of when and where both of these mycorrhizal types show up together or not in eucalypt systems. And then I'm going to spend the rest of the talk going over two case studies. So these are data that are largely unpublished. We've been doing this work over the course of the past almost a decade. And I'm currently taking advantage of the fact that, well, the invitation to give this talk was one stimulus to get back into these data, but also the fact that we're currently under lockdown and I can't do field work is really giving me an opportunity to finish up these data sets and hopefully have them published this year. And so these two case studies are looking at these two mycorrhizal types within root systems of individual eucalyptus plants, and then looking at how they co-occur in space and time in this in a broader grassy eucalyptus woodland. So, and then if you have any questions, any comments, I'm going to try to keep an eye on the chat while I talk, so feel free to put stuff in there. So the two mycorrhizal types that are relevant to eucalyptus systems are ectomycorrhizals and arbuscular mycorrhizals. Ectomycorrhiza, they differ in terms of their host breadth. They differ in terms of how they interact with their surrounding environment. I hope you can see my mouse as I wave it around. So ectomycorrhizal fungi are able to access inorganic and organic forms of nitrogen and phosphorus, whereas arbuscular mycorrhizal fungi are only able to access inorganic forms. And then transfer those forms to the host, and they don't have any sapotrophic capacity. And it's largely been this functional difference among these two mycorrhizal types that have made them the focus of a lot of recent work in ecosystem ecology, looking at the capacity of different vegetation systems that are dominated by these two mycorrhizal types to be able to accumulate and sequester carbon, including some fairly high profile papers in high impact journals. But they're largely considered to, vegetation is largely considered to associate with one of those two types. Another way of looking at this is this phylogenetic reconstruction of ancestral states. So this is a phylogeny of plants, and mapped onto this phylogeny are based on associations with extant taxa are expected states associated with their ancestors. As we've already stated, most hosts do form associations with arbuscular mycorrhizal fungi, and so those are the lineages in blue, and that's the ancestral state. So mycorrhizal associations are lost, not necessarily gained. There's several transitions to the ectomycorrhizal state, and then also some losses, several losses of the arbuscular mycorrhizal state. And the particular study that this figure is from, the modeling that they did suggests that there's this transition from lineages being arbuscular mycorrhizal to becoming non-mycorrhizal via a state where they're more facultative in terms of their associations. So they can be either arbuscular mycorrhizal or non-mycorrhizal. Now here at the bottom underneath the green line, I've got the Myrtaceae, and that's where Eucalyptus is. And so this is classified in this study and in many studies that try to contrast vegetation and ecosystems that belong to these different states as ectomycorrhizal. But we do know that arbuscular mycorrhizal associations can be observed quite frequently within Eucalyptus, and that we can actually see the functional structures, arbuscules, found in some of these roots. And so here are just a couple of examples. So one using Eucalyptus tatiana, where we see in an individual root system colonization by both an ectomycorrhizal fungus and an AM fungus, including several arbuscules in that root fragment that's colonized by the AM fungus. And then formerly Eucalyptus species where we see smooth ectomycorrhizal type associated with some roots, and then some arbuscules within the root cells of other root samples. So even though it's common for Eucalyptus to be classified in these synthesis studies as ectomycorrhizal, there are lots of examples where arbuscular mycorrhizas can be found. Now, there's been quite a bit of debate about what actually, what classifies, how do you actually classify mycorrhizal fungi as, or mycorrhizal associations into these different types. And so I just put up here a framework that's been very recently proposed by Francois Testa and others that gives a more solid approach to defining. So it's not just the fact that we're able to see structures that belong to both of these, but that the association should be functional. So we should see structures that are actually responsible for exchange of carbon for nutrients within those roots. And so for ectomycorrhizal associations, that would be a Hartig net or transfer cells, and arbuscular mycorrhizal associations, that would be arbuscules, whether they're the Paris type or the coil type, the arum type. Sorry, I got those reversed. And then it delves into a next level of that framework. So if we're observing both ectomycorrhizal associations and arbuscular mycorrhizal associations within the same species, do we observe them together in the same context? Or when we observe them, does it depend either temporally or spatially on the context in which either one of those is found or either one of those dominates? And so that's where the rest of this flow chart goes, where we might see, and well, I'll just jump into the cases that are relevant to eucalyptus. The data that are available suggest that these are context-dependent associations. So both might be present within the same species, but which one dominates is mostly dependent on the time or space in which these species are studied. So a couple of examples of these. So this study from Adams et al. That was focused on the North Queensland, looking at Eucalyptus grandis forests across several sites in North Queensland, and collecting roots from trees at these sites, Eucalyptus grandis trees, and looking for evidence of mycorrhization. And so that's the panel at the top here. And so what they found is that the vast majority of root fragments assessed were colonized by ectomycorrhizal fungi, and that there was evidence of root fragments colonized by arbuscular mycorrhizal fungi, but they were relatively infrequent, maxing out at about 25% of the root length. And that there were some sites where very few arbuscular mycorrhizal fungi were found associated with those trees. So this would be an example of a spatially dependent dual mycorrhizal association. And then there's also the temporal aspect of this. And so some studies, several studies in fact, have observed that arbuscular associations tend to be more common than ectomycorrhizal associations, or roughly equal in terms of their frequency when looking at seedlings. And so that's what was done here. So they planted seedlings into cores that were taken from these forests. And again, they assessed colonization by ectomycorrhizal fungi and arbuscular mycorrhizal fungi. They observed variation in the frequency of both, but a lot of variation in terms of the relative dominance of those types. And so again, in this case, it tended to be roughly even between the two types, or ectomycorrhizal fungi tended to be more abundant. Another example, so that's an Australian example. Here's a Brazilian example. So this is an example of planting eucalyptus seedlings and inoculating them with either a species of ectomycorrhizal fungus, so this was a Pizolithus species, or a species of arbuscular mycorrhizal fungus, and this was a species of Chloridioglomus. And looking at 5 different eucalyptus species. And although there was variation among the species, the patterns are roughly similar, where there's some nonlinearity in these associations. So the arbuscular mycorrhizal fungus was relatively slow to colonize. And so we started out with those roots being more so dominated by the ectomycorrhizal fungus. Then as colonization by the arbuscular mycorrhizal fungus took off, that dominance pattern switched, where it became more dominated by the arbuscular mycorrhizal fungus. But then colonization by that fungus slowed down, and colonization by the ectomycorrhizal fungus sped up. So that gradually it became more and more dominated by the ecto association. Some temporal succession occurring as these seedlings aged. That's another example of temporal context dependency. And then also show an example here from out in WA, which is a quite interesting example. There have been a few studies, including the one that I previously showed from Dos Santos et al., that have posed the hypothesis that some of this context dependency might be driven by negative interactions between mycorrhizal fungi of different types. And so we see here across a number of different sites where this eucalyptus species is found, a negative correlation in terms of the proportion of the root that is colonized by each type. So when arbuscular mycorrhizal fungi are frequently found within the roots, we tend to have low colonization by arbuscular mycorrhizal fungi, and vice versa. Now, there's another dimension to this, where we see that ectomycorrhizal fungi, their frequency of occurrence tends to be lower under canopies that are experienced in systems where eucalypt decline is being observed, and the canopy health is quite poor. And that we see the opposite pattern. So under conditions where we have a low health in the canopy, that's where we have low health in the canopy, that's where we tend to see higher rates of root colonization. And so one thought is that it actually might be this negative interaction between AM and ectofungi that's contributing to canopy to eucalypt decline in this system. But there's also the possibility that that this is actually related to other changes that are happening in the system as well. So this is just a photo that was taken from one of the sites that is experiencing quite a strong decline, and it's been invaded by a very dense mat of calla lily, which is a host for AM fungi. And so it could, something that also could be happening is that as the decline occurs and the canopy opens up and there's more light that's available to understory species that are like, many of these are hosts for AM fungi, that we're actually just seeing an increase in inoculum that's available to them. And so this is something that requires testing. Is this a direct interaction between fungi belonging to these different types, or is it more a symptom of other things that are happening within the ecosystem? And then kind of finally, in relation to this long introduction that I've been giving you, I've largely been dealing with these two mycorrhizal types as fairly uniform groups. But we do know that there is quite a bit of variation among species within these guilds of mycorrhizal fungi. And so just as an example, on the left, I've just posted this drawing that shows quite extensive variation in growth patterns, exploration types of ectomycorrhizal fungi, where in some cases you have some fairly smooth types. You don't have a lot of hyphae emanating from the mycorrhiza, and they don't form rhizomorphs. And then at the other extreme, you've got these long exploration types where you've got these rhizomorphs forming and extending long distances away from the roots. And that kind of intermediate stages, intermediate types, we have fairly dense hyphal mycelial systems that are growing in the area around the root system. And so these are, to a certain extent, related to species, but also to a certain extent related to the host with which that fungal species is interacting. But again, there's a huge amount of variation there. And then on the arbuscular mycorrhizal side, we've got variation among taxa in terms of whether they prioritize colonizing the soil environment or colonizing the root environment in terms of where biomass is being produced. And this, to a certain extent, is related to taxonomy. And so fungi within the Gygasperaceae in a few comparative studies have been found to allocate more biomass into the soil environment where they might interact with and be able to transport phosphorus to the host. Whereas there's this tendency more so in the Glomeraceae for extensive root colonization and still colonization of soil, but to a lesser extent than what's happening within the Gygasperaceae. So there's a need there to look at, well, okay, well, maybe we're seeing variation in terms of where these mycorrhizal types are being observed, but is that due to a complete turnover among those mycorrhizal types? Or is it more nuanced than that? Are we actually seeing shifts within these guilds that might be related to the extent to which they colonize the root versus the extent to which they produce a lot of mycelium within the soil? So just to synthesize that introductory part of this talk, we do know that many eucalyptus species can form both of these associations, and sometimes that does occur at the same time. But that the frequency with which the structures associated with these two types and how that changes through time can indicate the dominance. That in relation to the context dependency of whether dual mycorrhizal associations are actually forming within individual plants, or we're basically talking about the potential for these associations to form, and one of them is dominant at different time points in the life of a eucalypt, or in different locations where that eucalyptus species might be found. And most of the hypotheses that have been proposed to this point do invoke the benefit. So are these hosts receiving more of a benefit at a certain life stage from being associated with AM fungi? And then at another life stage being associated with ectofungi? or maybe it's related to interactions among these fungi within these groups. So that might be related to, for example, competition. So this is where we get into some of the original data that I'm going to be sharing with you today. So largely, not entirely, but largely in relation to, and in relation to the data that I'm going to be showing you, I'm going to be focusing largely on a grassy woodland system of grassy shale plain woodlands in the Cumberland Plain region around Sydney. And so this is, those are indicated in yellow on this map here. And so we see that pre-European settlement, there is quite an extensive distribution in the Sydney Basin of these Cumberland Plain woodlands. And that as a result of human activity, the extent of this woodland system has been greatly reduced. It's about 3 to 6% of that original extent. And so it is a threatened ecological community. There's a lot of work going into trying to protect and restore this vegetation type. And this vegetation type is quite interesting because it's got a mix of different mycorrhizal types within it. So typical of this vegetation type are 2 eucalyptus species, Eucalyptus malacana and Eucalyptus tereticornis. Both of these species are known to associate with ecto and AM fungi. And then these are grassy woodlands, so there's a lot of arbuscular mycorrhizal hosts. There's some acacia in these systems, and so we have this acacias are both AM and ecto, but they also form associations with rhizobia, and then some non-mycorrhizal taxa in there as well. We suspect that both AM and ecto fungi are beneficial to hosts in this, eucalypt hosts in this system. So here's an experiment that was done by an undergraduate student at the time, Beth Coleman, working with Jen Walker, looking at Eucalyptus tereticornis growth following inoculation with different combinations of ectofungi and arbuscular mycorrhizal fungi, and in different combinations. So different relative abundances of the inoculum of the different types. And so generally what we see is that it doesn't really matter whether they were inoculated entirely with the ectomycorrhizal fungi, entirely with the arbuscular mycorrhizal fungi, or with combinations of the two, that they were growing faster, at least in these seedling stages, relative to uninoculated plants. So that would suggest that these are actual, we're observing both arbuscular and ecto associations in these plants, but there's also some functional consequences associated with this. So that sets up the study system. All of the data that I'm going to be sharing with you today have been collected from our Eucalyptus face facility. So this is a free air carbon enrichment experiment. We have within our Cumberland Plain remnant that's located on our Richmond campus of Western Sydney Uni. Here's the part of the campus where all the buildings are. We have 6 of these rings, these arrays, that allow us to fumigate this woodland system with carbon dioxide. So 3 of these rings are set up as controls, and 3 of these rings are set up where the woodland is being fumigated with CO2 at a level 150 parts per million above ambient. There's quite a bit of kit associated with this experiment as well, and quite a bit of expertise, more importantly, quite a bit of expertise. And so we've got people that are looking at what's happening in the soil, including soil biological patterns, but also soil chemical patterns. And then people that are looking at what's happening in the understory vegetation, and people looking at what's happening in the canopy. Some of the findings that we've had from this experiment that are relevant here. What we see is that elevated CO2 leads to increased carbon fixation within the trees. So photosynthesis is happening to a greater extent. This is not translating to increased productivity in the trees. And so most of this carbon is actually being lost from the ecosystem by soil respiration or respiration from soil. We're seeing some compositional turnover in the understory vegetation, and that we're seeing reduced C4 cover. And that early in the experiment, but this didn't necessarily persist, although we are following up on that. We're seeing increases in availability of nitrogen and phosphorus under elevated CO2, which is possibly related to increased turnover of soil microbes. It's been possible, so this was work led by Ming-Kai Zhang. We were able to construct a carbon budget associated with the various data sets that have been collected at the site. So I'll just point you to this box here. So we see that there's not really much change in terms of the amount of carbon that's found within the mycorrhizal pool in this system. And that mycorrhizal pool is relatively small compared to the other carbon pools in the system. So it's about 1/10 of the fine root pool, and about 1/100th of the coarse root pool. But through an exercise, modeling exercise that Ming-Kai did, was trying to find out where is this carbon traveling through the system. And this is not something that was measured. It's something that's estimated from the model. So it'll be really interesting to verify it. But what the model predicts is that the carbon that's flowing through the system, the path through which it's flowing, is a large chunk of that is through this the mycorrhizal system. And so what these numbers here mean is it suggests that the turnover of carbon within that mycorrhizal pool is more than twice as much as what's observed in the under ambient CO2. And so again, these are predictions from the model. It's something that does need to be validated, but it is quite interesting, and it leads to hypotheses about, okay, well, if this is something that's actually happening, perhaps we should be seeing changes in the hyphae that are being produced by mycorrhizal fungi in the system, whether that's as a result of changes in the composition of species that are present, or changes in the traits associated with within fungal species that are leading to this increased turnover of mycelium. Anyway, so getting back to these shifts that are occurring, or that are hypothesized to occur, our original hypotheses going into this study were that, one, simply that this shift does occur, that as trees age, we see them predominantly colonized by arbuscular mycorrhizal fungi, and then we see a shift in dominance towards ectomycorrhizal fungi. And then the second hypothesis is that if the shift does occur, the timing of this shift should depend on the physiology of the host. So changes in, for example, nutrient availability under elevated CO2 relative to ambient CO2 should lead to a shift in the timing of when this shift in dominance occurs. If the main determinant of whether one of those two mycorrhizal types dominate depends on what the host is getting out of the association. And then the third hypothesis is that this shift oversimplifies. Just looking at these mycorrhizal types, it oversimplifies what might be happening within these guilds. And so the example posed here is that we actually see AM fungi present at all times of the life of the host. But what we're actually seeing is a shift towards more of these fungi that tend to colonize the root to a lesser extent. So in order to test these hypotheses, what we did was, so this is work that was led by Jen Walker. So Jen so the idea was that we would take soil cores, instead of planting seedlings out into the field, where they would have a very low chance of surviving. She took intact soil cores in order to attempt to preserve the inoculum that was present within that soil, planted seedlings, Eucalyptus tereticornis seedlings, into those cores, and then after 4 months took those cores and put them back in the same place that she collected them from initially. And then over the course of the next year, at 8, 12, and 16 months, after planting those seedlings, harvested those plants again. And so these cores were collected and then reinserted around individual Eucalyptus tereticornis trees, so within a meter of those trees, out within those face rings. And so looking at some of what Jen found when she collected these plants again. So initially, we had about after 4 months, those roots were about 50% colonized by ectomycorrhizal fungi, and then were less colonized by arbuscular mycorrhizal fungi. So between 10 and 30% of the root length was colonized. As the experiment progressed, we saw an increase to the point where after 8 months, they were about 85% colonized by ectos and 100% colonized by ectos after 12 months. And then it eventually leveled off at about 50% colonized by AM fungi at each of those subsequent harvests after planting back out into the field. So in terms of effects of elevated CO2, we saw more rapid colonization by that in that initial phase as we went from 4 to 8 months, where, after 8 months, it was almost 100% of roots were colonized under elevated CO2, which suggests that given that this was occurring at some point after they were planted back out into the field, that suggests that, well, maybe this is a host physiology effect. Whereas the effect of elevated CO2 was observed in that initial 4-month phase, where, as I should say, I didn't say it before, in that phase, they were being grown under ambient CO2, so that they weren't being grown in these cores out in the rings. They were taken to another place on the site and grown. So this would reflect variation among elevated and ambient CO2 in terms of the inoculum that was present. So it suggests that there was more AM fungal inoculum that was present in those soils under the elevated CO2 treatment. We did some Illumina sequencing of roots in order to get an idea about which fungal taxa were present associated with these roots. And so these aren't all the taxa that we observed. I just chose a subset of taxa to give you an idea about what are some of the temporal patterns associated with these. We saw an increase in Amanita, consistent increase in Amanita over the course of the entire experiment. There was variation in some of the other taxa. We observed Leucaria early on, but then that quickly, after about 8 months, that disappeared. Whereas Physolithus, it took 8 to 12 months before that was actually starting to show up on the roots. And then Roulandiella was, there was variation in terms of how frequently we observed it, but it was observed at all time points that we were studying. We also collected soil samples to get an idea about what was available for inoculum in that soil pool. And this, for some of them, it kind of matched what we saw. So we saw where we had more when we had more inoculum associated with Roulandiella, we tended to see higher rates of colonization, at least based on the sequencing data. To a certain extent, we saw a similar thing with Amanita, although there was that relationship falls off when we're looking at after 16 months. And then Leucaria and Physolithus would seem to be very independently of what was present, what was detected in that soil inoculum. I mentioned that we also were collecting roots associated with, I mentioned that these cores were put back out around mature eucalyptus trees. And so where we could, we also attempted to catch roots that were coming from that eucalyptus tree that were also passing through our soil samples. And so we were able to do that for 2 of the 3 time points at which these cores were collected from within the rings. And we saw some correspondence when it came to, for example, Leucaria, and to a certain extent Physolithus. So where we observed these fungi associated with those mature roots of the trees, we were also detecting these taxa within those seedling roots. So that suggests that perhaps this isn't necessarily a successional thing. It's more an issue of, again, inoculum. The fact that the trees these mature trees had were colonized by these different mycorrhizal types, or these mycorrhizal species, meant that they were present and able to colonize the roots of the seedlings as well. Now I'll skip to what we observed with the AM fungi, again using amplicon sequencing. And again, we saw, these are not necessarily all the taxa that we saw. These aren't all the taxa that we saw, but there's some examples that show variation. So again, we saw Chloridoglomus fairly early on, but then that disappeared. And in those time points, we saw species belonging to Glomus. We saw increases through time when it came to Gigaspora, but then that completely disappeared at that 3rd time point. And then again, this gradual increase of Rhizophagus. And I won't show it because I'm running short on time. But again, we saw variation in terms of whether this matched up with what we were also finding in the soil and what we were finding in association with the mature roots that were growing through these soil cores. Now, I'm going to jump ahead a few slides because we are running short on time. And I just want to show this result. So this is a chord diagram on the right-hand side here. And so the data in this represent residual correlations. So these are correlations in terms of where in what abundance taxa are found after accounting for environmental drivers. So things like soil chemistry, things like whether the rings were fumigated or not, things like traits associated with the plants that they were interacting with. And what we found is that we found a lot of evidence. We found evidence for positive interactions being observed among mycorrhizal fungi. And so these are the brown ribbons that connect the different taxa. We also observed evidence of negative interactions among these species. And so these are the yellow ribbons, but that it was more dependent on the taxa that were interacting and not dependent at all on the interaction type. So the frequency of interactions, whether they were negative or positive, was roughly the same when we were comparing ectomycorrhizal fungi interacting with other ectomycorrhizal fungi or ectomycorrhizal fungi interacting with AM fungi. So competition, facilitation, these types of interactions might be occurring, but it's probably more nuanced than just replacement of one mycorrhizal type by another mycorrhizal type. So just a summary of this first case study. We didn't really see a shift in AM and EM fungi over the course of this experiment looking at these seedlings, and they tended to both accumulate. And they accumulated independently of each other, and that was dependent on the environment. We did see compositional turnover within these guilds, and we did see interactions among taxa within these guilds. But again, these tended to be less related to what was happening in the other guilds and more related to what was happening within guilds. So less of a mycorrhizal type interaction. I don't really have much time left to get into the second case study, which I was for those of you who have seen me talk before, I do tend to spend a lot of time on previous slides. I was trying not to do that this time, so I apologize. But what I was hoping to show you here is that we've got a really rich data set, and I'll jump ahead to a couple of slides. We have a really rich data set of, again, using fungal sequencing data to get an idea of changes in relative abundance of mycorrhizal fungi belonging to ectomycorrhizal or arbuscular mycorrhizal taxa along almost a decade of soil samples collected under very, very different environmental conditions. So here the arrows indicate where we were collecting soil samples, and the points and the lines represent soil moisture throughout the extent of this experiment. And these are data that are collected at a ring level. We also observe variation in things like canopy health. So we've got quite a bit of variation from year to year. So these are images that are taken at security cameras within the rings. And it gives you an idea about how leaf area index can change quite extensively. And for one period, over the course of 1 year, this was we had a very large drop in canopy cover as a result of a infestation of psyllids, cardiospina psyllids, that led to almost complete defoliation across the entire site. And so we took this opportunity to look at relationships between some of these environmental conditions and the relative abundance of these different mycorrhizal fungi, or their probability of occurrence. And so the thing that explained the thing that was most important in determining the abundance of these different taxa was canopy cover. And so this kind of corresponds to that eucalypt dieback example that I started off that I had early on in the talk. And so as canopy cover increases, we get a decrease in our detection of arbuscular mycorrhizal fungi. And that, to a certain extent, it's marginal in its non-significance, but to a certain extent, that is less of a decline in the detection of AM fungi under elevated CO2 compared to ambient CO2. And so this would be consistent with a canopy that's opening up, that is meaning that there's more light available to the understory, and that those understories are serving as hosts for arbuscular mycorrhizal fungi. And then under those same conditions, that's what's happening over at this end of the plot. Under those same conditions, because we've got very poor canopy associated with the host trees for the ectomycorrhizal fungi, that's where we see reductions in our ability to detect them. And that as canopy cover increases, we detect ectomycorrhizal fungi more frequently, and that slope of that relationship is actually higher under elevated CO2. So I won't show you some of the other patterns. Instead, I'm just going to jump to the conclusions. So in these systems that we've been studying, and actually other systems that we've been studying as well, we're observing functionally diverse partnerships within roots. Whether they're ectomycorrhizal fungi or arbuscular mycorrhizal fungi or both, we are detecting functionally diverse partnerships in relation to the types of fungi that we're finding in association with these plants. There are interactions there. They tend to be interactions more so when it comes to the individual taxa within those groups, and less so between arbuscular mycorrhizal fungi and ectomycorrhizal fungi. And that those range from positive to negative. However, and I didn't show these data today, we do have some data sets that suggest under certain conditions, the relationships between those different mycorrhizal types might actually be stronger. And so maybe I'm hoping to be able to share that with you fairly soon. Both AM fungi and ectomycorrhizal fungi are dynamic. For AM fungi, they're associated with canopy cover, and I didn't show you, but they're also associated with soil moisture. And then ectofungi are dynamic, but they're primarily associated with canopy cover. And that there's spatial and temporal environmental variation that leads to variation in inoculum loads, and that's this variation in inoculum loads, I think, that is supporting, at least in the woodlands that we've been looking at, these context-dependent dual associations, and less so the actual needs of the host plant. Okay. And then I'll just throw this slide up here. Maybe we can use this as a just as a teaser for some discussions that maybe we could have before the Zoom session ends. But I also need to acknowledge everyone that helped out, and the funding agencies that were that supported most of this research. Thanks very much. I can't hear you, Tracy.
Sorry, I hope you can hear me now.
Yep.
Okay. These are the just in case headphones. Okay, so thank you so much for that fascinating talk. I'm sure we would love to see what the results are of your case study and the future directions. We've got already one question up, so I'll go ahead and ask that. Renee Johansson asks, in regards to case study 1, mycorrhizal fungi are also known to vary with season. Do you think this might explain change over time as well as plant age?
There's certainly seasonality in the abundance and the detection of mycorrhizal fungal species. And I was expecting to see more of a seasonal pattern. We didn't actually detect much of a seasonal pattern when it came to when it came to this particular woodland that we were studying. I was more strongly related to when rainfall was occurring, again, issues, extent to which the canopy was closed or open, which was largely related to rainfall, but also related to the abundance of herbivores in the canopy. And then, yeah, so I wouldn't, there were temporal patterns, but I wouldn't call them seasonal patterns, in that they weren't really consistent from year to year. They were consistent in terms of when particular environmental conditions were showing up.
Okay, thank you. I have a question. In the transplant experiment, did you notice a difference in the health of the plants under the different treatments?
Not so much in the health. Maybe to a very small extent, the growth rates, but it was those data were fairly noisy. So the seedlings were growing at roughly the same rate under the 2 CO2 conditions. Any variation in growth rate was probably more linked to nutrient availability, which at the site, it's quite an interesting site, because there's quite a bit of variation in terms of the soil environment. So we've got, there's alternating sand and clay layers. The depth of the clay layer varies across the site, and the sandiness of the soil varies across the site as well. And so we've got a gradient in nutrient availability at the site too. And that was more of a driver of seedling growth than the CO2 condition.
Great. Renee is just responding to your previous answer, saying, interesting, thank you. Are you going to look at or publish on other endophytes?
It's a very rich data set. While I was preparing some of these slides, I was looking in more depth at some of the other taxa that are present within these soils. And so we do have some endophyte taxa that are detected within the roots of these. And there are some pathogenic taxa in there as well. So I can see myself being very busy
for a while.
But I need to focus. I need to get this stuff published. So I need to just focus on the mycorrhizal stuff at this.
Well, we look forward to what happens. Unfortunately, we have to end up now. I've just put up an announcement about our next talk. Kentaro Hosaka from the National Museum of Nature and Science in Japan will be speaking at the end of next month, same time, same place. So feel free to join us for then. For now, Jeff, thank you so much for joining us for a fascinating talk. I do have another question from Tom May up here, so if we'd like to answer that right now, we can do that. For anyone else, thank you so much for joining us. I'll go ahead and ask this question from Tom quickly, and after that, we'll just end up. So Tom says, thanks so much for the fantastic talk. Thinking about optimum time to sample ECM communities, such as by eDNA, how much does the variation with season and weather affect the diversity? What would you recommend as best time for sampling, apart from trying to sample all sites at the same time?
Yeah, what I would recommend is not necessarily seasonal sampling. So I guess it depends on the aim. If the aim is to try to maximize our capacity to be able to detect the most ectospecies, in this system, what I would recommend is if it's dry, don't bother trying. Like, it's the same as if you were going out looking for mushrooms. There's not really that much point in going out and collecting and doing surveys if it's hot and dry. Our capacity to be able to detect fungi in the system was reduced under those conditions where the soil was dry and the weather was hot. And that's possibly partly due to reductions in the abundance of these, but we also found that under those conditions, the sequencing data that we were getting back tended to be dominated by saprotrophic fungi. So even though those ectofungi might have been present in those samples, our ability to observe them in the sequencing data was reduced because of this, probably the relative pool of DNA associated with saprotrophs was just so much greater. So I would not necessarily focus on seasons, I would focus on environmental conditions.
Okay, great. Thank you so much, Jeff, and to everyone else for joining us. We hope to see you all next month for our next seminar. And don't forget to answer the short survey at the end of this seminar. Thanks again, Jeff.
Thank you, Tracy. Thanks everyone for listening.